Alright, everybody, welcome back to lecture 16 here. We're going to be talking about nucleotide catabolism and some clinical aspects of the metabolic pathways for these things. This is a shorter lecture, actually, so shouldn't be too bad, but, you know, the nucleic acid turnover is just like process of producing and degrading these different nucleotides that we've made. Helpful for, signaling pathways, so like gene regulation, all that stuff. And, salvage pathways are important because they help with maintaining the nucleotide pool. And they also just like stop us from having to make new nucleotides because nucleotide synthesis is not very efficient. So, yeah. So here we're going to talk about the degradation of purines. So, basically, we have a bunch of endonucleases and exonucleases that help cleave DNA right into nucleotides. We've talked about this before. And then there's these enzymes called nucleotidases that cleave the phosphates to yield nucleosides. And nucleosides, remember, are just the sugar plus the nitrogenous base. And these nucleosidases use water, and this should make sense. It's just going to be a transesterification reaction where you basically attack the phosphate and you kick it off. And so adenosine can be cleaved to, adenine, or undergo deamination to inosine, which you guys have talked about. And, hypoxanthine is going to be the deamination product of adenine, which we'll talk about here in a second. So you can see here we've got two different pathways. Adenosine, here we got the sine at the end. Usually it refers to when there's no phosphate groups on. So we have GMP and AMP here. You use a 5' nucleotidase. It's 5' because of the phosphates on the 5' end. And then you have adenosine here, and you have guanosine here. And so what you do is you use a nucleosidase, right? Because now it's a nucleosidase is a bit different from a nucleotidase. A nucleosidase is one that cleaves the sugar from the base, and so you release a ribose. Bless me. Sorry about that. A nucleosidase is one that cleaves a nucleoside, so it like cleaves this bond right here, if you want to think of it like that. And then you end up with this guanine nucleotide here. Or in the case of inosine, what ends up happening is you use adenosine, or sorry, not in the case of inosine, in the case of adenosine or adenine, you go and you get adenosine, you use adenosine deaminase, which removes this NH3, and then you use the nucleosidase. So basically you're removing the N from the adenine, and then you're turning it into hypoxanthine, which is this guy here. And so then we move on to the next aspect of this pathway is, we've got our guanine and our hypoxanthine, which we just made from, our adenine after we deaminated it and cut it off with a nucleosidase. And what's going to happen here is that we're going to use xanthine oxidase. And what xanthine oxidase does is it puts an extra OH onto this hypoxanthine we just formed. And for guanine, all we have to use is just use a guanine deaminase, because guanine already has this NH2 group in front. We just have to like transaminate it, right? Just remove it, replace it with an H2O. So that's why you don't need this whole extra xanthine oxidase step for guanine. And so you make this xanthine, right? So this is hypoxanthine because it's missing an OH group or an O group. And this is xanthine because it's got the extra OH, and then you use xanthine oxidase again to add an extra OH to it, and this is now uric acid. But yeah, this is I believe is secreted in your urine, actually, if I'm not mistaken, as well. So uric acid represents nitrogenous waste product, and we secrete 0.6 grams of it per day. But yeah. And the thing is, if you have excess uric acid, it actually causes gout. So uric acid is not very soluble. It's a pretty aromatic compound, right? There's a couple OH groups on there and maybe some hydrogen bonds, but it's pretty there's some hydrophobicity to it. So a lot of it, too much of it, can definitely lead to like clogging up and stuff like that. And so you can also have it crystallize with sodium in the body to make sodium urate, and this ends up precipitating in the kidneys and in your joints, and it gets really, really painful. So the treatment for this is a combination of changing your diet and drug therapies. So like an example of allopurinol is a substrate of xanthine oxidase. And basically what we're doing is we're actually trying to stop the production of hypoxanthine into xanthine, and then uric acid, right? So when we make this oxypurinol, and this oxypurinol comes back and inhibits xanthine oxidase, basically. So yeah, when you secrete it, you're actually going to be secreting hypoxanthine, and you'll secrete xanthine as well. Yeah, I'm trying to remember the exact but yeah, basically you're gonna make a bunch of like this xanthine and hypoxanthine, and you're not gonna make like any uric acid really, and so you're gonna secrete out the hypoxanthine instead, and maybe some xanthine as well. I'm trying to remember. I can't remember the exact makeup. I believe I'm trying to remember if xanthine oxidase has 2 different binding sites for hypoxanthine and xanthine. I can't quite remember though. So yeah, unfortunately I can't remember that off the top of my head. So adenosine deaminase deficiency, this is just if you don't have any adenosine deaminase, right? So if you lose this, you can't really make inosine. So accumulation of adenosine and deoxyadenosine leads to higher levels of ATP and DATP, and so what that does is that turns RNR off, remember, and so that would disrupt the pool of DNTPs, and so DNA synthesis and repair is messed up a bunch. And so there's a whole bunch of other things, like so it causes a severe combined immunodeficiency disorder, and these B and T lymphocytes aren't able to proliferate. They're not able to like replicate and stuff like that because this adenosine deaminase is broken, and so we're not able to generate nucleotides, and so we're gonna die that way. So we have hypoxanthine guanine phosphoribosyl transferase. This is a enzyme that it's another genetic disorder enzyme. Basically, it's a disruption of the salvage pathway. And it results in the overproduction of purines by the de novo pathway, and it does that because this enzyme here breaks down. So for example, if you have guanine, right? Normally, what you can do is you can use this HGPRT to attach it to a sugar, and then just make GMP. But if you break the enzyme, you actually can't do that anymore, and so if you can't do that anymore, you have to like de novo synthesize everything, and that just takes up so much energy. And then what that means is that this guanine is just going to be thrown out, and it's going to be turned into uric acid, and so you're going to get like gout-like tissue and a bunch of swelling, because remember, we can't reuse this anymore because this enzyme doesn't function anymore. That also is true for hypoxanthine, which you could technically attach onto a ribose and make it inosine, and then if you wanted to do some sort of deamination reaction to turn it into adenine, you could. But if this enzyme is broken, this just is just going to get thrown out, and again it's going to turn into uric acid, and you're going to get gout again, and that process all over again. So okay, so for catabolism of pyrimidines, when we break these down, we usually result in the formation of acetyl-CoA and succinyl-CoA. And then we also produce some NH3/NH4+, which is incorporated into the urea cycle. And urea is like the one that's really in urine. Very, very soluble in urine, so you'll see the structure of it soon. It's very easy to get rid of. So we can see here, I wanna talk about this real quick, but like I don't know if you have to know the exact ins and outs of this. Ah, yes, they do talk about it a bit more down here, I think. But if we look here, we can see that, you know, we can remove this nitrogen to turn the cytosine into uracil, right? And then this nitrogen just kind of gets added to urea. And so what we do then is we use the carbons from uracil and from thymine to make acetyl-CoA and succinyl-CoA. And this extra carbon that was added from THF is released as CO2, which can be used to make urea, basically. So it's like, you know, this carbon here is this carbon, if you want to think of it like that, and then the cytosine, this N is this NH2 here, and so we make urea, succinyl-CoA, and acetyl-CoA that we can then use in our citric acid cycle and stuff like that. So, targeting nucleotide metabolism. So, the majority of adult cells are in the non-dividing phase, so they're in G0. But cancer cells, right? Cancer cells are cells that are constantly dividing, and so that should scream to you, wait a minute, what if we, you know, use a drug to inhibit things that are active when cells are dividing? Because cancer cells are dividing a lot, and so if we preferentially target enzymes that are active during DNA replication, that'll be great, because that means that they would be inhibited in a lot of these cells, because, you know, most of your cells are not dividing anymore. So that's kind of the main point of this. So chemotherapy, pharmaceutical agents, they're designed to prevent the growth of these cancer cells by targeting enzymes that involve the nucleotide metabolism. So here is an example of that, targeting thymidylate synthase. So thymidylate synthase is the one that makes DTTP. So thymine, obviously, you know, if you don't have a dividing cell, thymine is not very useful. So, but if you have a dividing cell, it's very necessary. So cancer cells, we can target them through drugs like this. So thymidylate synthase catalyzes DUMP to DTMP, and so we can use this 5-fluoro uracil structure. Basically a uracil analog. We just add an F to the carbon over here, and this is taken up by cells and converted into FDUMP, and this FDUMP is actually an irreversible inhibitor of thymidylate synthase. So it's converted to this by RNR, but then this actually binds to thymidylate synthase and blocks it from making blocks DTMP from being made. Because it blocks DUMP from binding to thymidylate synthase, so it blocks DTMP production. And so what ends up happening is that this fluoro-UTP actually ends up being incorporated into DNA because there's no thymine, and so it leads to double-stranded breaks and disrupts all the functions of the DNA and ends up killing the cancer usually. And so, you know, here's an example of what's going on here. Basically, like, this enzyme is broken, right? And so this enzyme is broken, there's no TMP being made, and so therefore our cells cannot use TTP. They use uracil instead because they need to somehow fix their DNA, and that actually ends up breaking them even further. Alright, dihydrofolate reductase. So dihydrofolate reductase is another thing that you could target. So remember that for dihydrofolate reductase is important, and because we use it to make, we use THF, right? And THF is used to add this methyl group onto uracil to make thymine. So, yeah, it's very important in that regard because it's used to add the methyl group to thymine. If you don't have it, you can't make thymine. So you could, what happens with this enzyme is dihydrofolate reductase. It catalyzes the conversion of folate, which is what I talked about before with women in pregnancy, remember? Why women tend to take folate supplements. So you take folate, and it takes folate, turns into dihydrofolate, and then turns into tetrahydrofolate, which hopefully should make sense. It's a reduction because you're adding H's on there, and H's, you know, they add extra electrons. So you can see here, we initially have only one H, then we have two H's, and then 4 H's. So this is tetrahydrofolate. And so that tetrahydrofolate actually allows us to donate a methyl group onto the a methyl group onto our UMP. So that's why that's important. And if you break this enzyme, you actually can't do that anymore. So, yeah. So the drug we use for that is methotrexate. Oh my god, I always mess this one up. Methotrexate. Yeah, so methotrexate is an analog for folate and dihydrofolate. It's an antagonist of folate. So basically, it's a high affinity competitive inhibitor of the dihydrofolate reductase, and basically it starves them of DTMP, similar to what we did with allopurinol, right? And not allopurinol, similar to what we did with the 5-fluorouracil, right? We starved the cell of TMP, and so it can't replicate. Yeah, so the only thing that's like a bit dangerous about using the dihydrofolate reductase stuff instead of like the 5-fluorouracil stuff maybe is that this disrupts purine synthesis as well. But yeah, I'm trying to see if they discuss that. Vitamin B9. But yeah, just remember like if you remember, de novo purine synthesis, right, uses THF in forming the actual backbone structure of the nitrogenous base. It's like 2 of the carbon atoms. Like remember, I think the bottom 2 nitrogens come from a glutamine. Remember, and then the 2 carbons to the right and left of those come from THF. So if you actually break that, then you can't actually make new purines. So that's actually a problem. So it's not the best to use that because, we still need to make normal purines. We just don't need DTTPs, right? Especially if our cells are not dividing. So that's why I would say that using a thing like 5-fluorouracil is probably a better option than using something like this, because this also affects your normal cells. Because just to clarify that as well, if you use this and disrupt purine synthesis, you don't make ATP, and ATP is just very important for all your cells, right? You can also target RNR. There are different things that you could use. So hydroxyurea is one of them. It's a 1-electron reducing agent. It's basically a radical scavenger, and it gets rid of this free radical, which breaks the entirety of RNR. So that's another option you could use. And yeah, that's actually pretty much it for this video. These are cool little case things. They're obviously you should study them, but I don't think there's going to be like more than a couple questions on this if I had to say. But that's not to say that it's not going to show up on the exam, period. Make sure you study it. Make sure you keep it in mind. And yeah, hope this video was helpful. If you have any questions, be sure to stop by the IC or leave a comment on it. And yeah, have a good day and happy studying.